For my project, I focused on the glycolytic pathway, but more specifically, the highly regulated irreversible third step, which is when fructose 6-phosphate is turned to fructose 1,6-bisphosphate and catalyzed by the enzyme phosphofructokinase. This is the committed step of the glycolytic pathway, meaning that the pathway is committed to finishing glycolysis and making pyruvate, as well as ATP and NADH. It is also regulated and irreversible due to its highly negative delta G. Another important note is that the reaction does require ATP to proceed. Here is the mole star model for the inactive or T state. PFK's quaternary structure is a homotetramer, meaning there are 4 identical subunits. One here is shown in pink, another in purple, the third in brown, and the fourth in green. Phosphofructokinase also has 4 identical active sites as well as 4 identical allosteric sites. There is one for each subunit. Here shows an example of an active site on one of the subunits. Fructose 6-phosphate and ATP as well as magnesium bind at the active site. Here we can see an ATP molecule in the active site as well as the magnesium, and found here we can see the fructose 6-phosphate molecule. This subunit also shows the regulatory site found here where ATP is currently bound. ATP and AMP compete at this allosteric or regulator site, which I will talk about later in the video. This shows the simplified mechanism of phosphofructokinase, where phosphofructokinase transfers a phosphate molecule from ATP to fructose, which creates fructose 1,6-biphosphate. In doing so, controls entry into the pathway. Magnesium is also used in glycolysis whenever an enzyme is transferring a phosphate group to or from a molecule. The reason for its use as a cofactor is to shield the negative charges from the phosphate group of ATP to allow the enzyme to properly function. This image shows the same mechanism as before, but in the active site of phosphofructokinase, with some of the important amino acids that are involved. The oxygen from aspartate forms a hydrogen bond with hydrogen from fructose 6-phosphate. Another oxygen from aspartate, as well as 2 phosphates from ATP, form ionic bonds with magnesium. The bonds that are formed with the amino acid residues stabilize the molecules to be able to undergo the reaction. The arginine residue also forms a hydrogen bond with phosphate from ATP and stimulates the cleavage of the phosphodiester bond and transfer of the phosphate group to fructose 6-phosphate to form fructose 1,6-bisphosphate. After the reaction catalyzed by phosphofructokinase takes place, this is what the active site looks like, with ATP molecule and the fructose 1,6-bisphosphate and all of the amino acid side chains that interact with these molecules. Here shows the actual phosphofructokinase enzyme with some of the active sites present. We can see that there is a fructose 6-phosphate molecule here. The green ball at the top is represents the magnesium, and one of the important amino acids we had previously mentioned, arginine, is bound to the phosphate on fructose 6-phosphate. Up here, we can also see the ATP molecule that is bound to the magnesium. Regulation happens at the allosteric site on the enzyme. Phosphofructokinase is regulated by being allosterically inhibited by ATP and stimulated by AMP. There is an R state, which is the active state, and a T state, which is the inactive state. Fructose 6-phosphate binds with a high affinity to the R state, but not the T state. For every molecule of fructose 6-phosphate that binds to PFK, the enzyme progressively shifts from T state to R state. ATP is shown here currently bound to the allosteric site, but AMP, the allosteric activator, can also bind to the allosteric or regulatory site to facilitate the formation of the R state. AMP gets rid of ATP's inhibitory effect because AMP signals that more ATP needs to be made. ATP, the inhibitor, binds to the site also to form the T state, which inhibits enzyme activity. This means that if there is an abundance of ATP in the body, there is not a need for the body to make more, and therefore ATP inhibits phosphofructokinase as a whole by decreasing its affinity for fructose 6-phosphate. Phosphofructokinase deficiency, also known as Tori disease, is a rare muscular metabolic disorder with an autosomal recessive inheritance pattern, where people are intolerant to vigorous exercise, and more specifically, it interferes with the processing of food, in this case carbohydrates, for energy production. The mutation impairs the ability of phosphofructokinase to phosphorylate fructose 6-phosphate, like we see here. Inhibition of this step prevents the formation of ATP from ADP, which results in a lack of available energy for muscles during heavy exercise. This results in muscle cramping and pain that are common symptoms of the disease. This is another figure showing Tari disease, where muscle PFK and red blood cell PFK are completely absent, and therefore glycolysis cannot proceed. PFK is an extremely important enzyme for the function of the glycolytic pathway and overall creating fuel for the body, and has serious implications when deficient.